Okay, development of male and female reproductive systems. So this is like embryology, but just of the reproductive system from the genetics. So starting with we've mentioned before when we talked about gametogenesis, 23 pairs of chromosomes, 22 of them are autosomal. The last pair is where we see the sex chromosomes, so the X or the Y. Females inherit an X from both the mother and the father gamete, whereas males inherit the X from mom and the Y has to come from the father. So essentially, that during development, the males are responsible for determining the sex of the offspring because they can donate either an X or a Y sperm. On the Y chromosome, there's only about 15 genes, but the one that seems to be important for creating maleness is one that's called SRY, stands for substance releasing Y chromosome. It's only active for a few days, around 15 days after fertilization, and that's the trigger for maleness. What we see early on in development is that both male and female develop into this kind of sexually indifferent embryo. And so what that means is they have a generic gonad that forms near what's going to become the kidneys. There are some generic structures that are going to go on to become the genitals, but every embryo forms with both the Mullerian and the Wolffian ducts, and the Mullerian ducts could become the future female ducts, and the Wolffian ducts can become future male ducts. So the default pathway is for the Mullerian ducts to make full connections and for the embryo to become female. SRY is what triggers the development of the Wolffian ducts and the formation of the male tubes. And this happens very early in development. It's one of the kind of early onsets in terms of determining what's going to happen with the embryo. So what we see here is early in embryonic development. So just to run through these structures, there's this gonadal ridge, and that's what ends up being either the testy or the ovary. And that is surrounded by the mesonephros, which is going to become the kidney. And then there's two sets of ducts. So you can see around the outside, this slender pink duct, which is the Mullerian duct. Notice it's not directly connected to the gonadal ridge, just like the fimbriae wouldn't be directly connected to the ovary. Whereas coming from a separate location, you see the development of the Mullerian ducts, which are going to end up being much more closely connected to that gonadal ridge. So the question is, what happens whether there's SRY or whether there's not SRY? So if SRY is present, you see the development of, and you can see the tube develop very well into the structures that are going to be the ductus deferens connecting to that gonad. And then the connections end up being direct, so that once sperm is produced in the testy, it will end up in the epididymis. One thing that would have to happen is that this is still up in the abdomen, so this testy has to be pulled down out of the abdomen into the scrotum. But what would happen is you'd see the withering of those Mullerian ducts. Now, one of the things that can happen is even if this development takes place, if the testy doesn't descend out of the abdomen, so it creates condition undescended testy, that testicle becomes feminized. Feminized meaning it doesn't produce sperm and it doesn't produce androgens, and so you would have a sexually indifferent individual. If SRY is not present, the default pathway is that those Mullerian ducts are going to be the ones that develop and the Wolffian ducts disappear. And at the tips of those Mullerian ducts is where you see that open part of the oviduct that's going to form the fimbriae that will attach over to the ovaries. Now, in terms of the genitalia, again, the initial phase is indifferent. And so these are color-coded as homologous structures. So the three color codes you see would be the genital tubercle, you see the labial scrotal swelling in yellow, and then you see this urethral groove in pink. And so if we follow these through the phases, if we see male development, that genital tubercle ends up extending and forming the erectile tissue that makes up the glans and the shaft of the penis and the skin around that. The labial scrotal swellings end up fusing and forming the scrotum, trapping that urethral fold inside the penis, and that creates that internal urethra inside the penis. And then one of the things we ask you to look for in lab, it's very difficult to see on older donor, is you see that formation of the raphe along midline, which is that fusion site. In females, there's no fusion. So we go back to this original indifferent stage. And what we see is that genital tubercle, instead of growing outward, ends up growing just in position at the anterior portion of the urogenital opening and forms the erectile tissue, that's the glans clitoris. The labial scrotal swellings, instead of fusing, form labia majora, labia minora, and then in the middle, that urethral fold ends up staying patent or open, and that creates the space where you're going to have the opening to the urethra and then the orifice for the vaginal canal.